Dear first year students, I hope you are all fine with all your activities. As you know that we are discussing about the embryology and we have finished some important aspect, special reference with development of male-female gametophyte, then pollination, then post-fertilization changes, that is double fertilization, after that formation of embryo. Now we shall proceed towards another important aspect, and it is called as a double formation of endosperm. Endosperm is the most important characteristic of the angiosperms, and you are supposed to remember that in this particular way, this endosperm formation takes place, where you will find that initially this fertilization, double fertilization takes place, and in that double fertilization, you will find that these angiospermic plants that undergoes division redivision process and form the zygote. The zygote later on divide, redivide, and form the embryo that we have discussed a lot. Later on, what's happened? This triple fusion, which is present toward the lower side, that undergoes initially mitotic division, later on meiotic divisions, and then it goes divide and redivide and then form a particular complex of tissues, and that complex of the tissues where the large quantity of the cells are present, it is called as the endosperm. Now this endosperm formation takes place in three ways. The first one is the nucleus, where only the nucleus divide and redivide and form new endosperm. It is called as a nuclear endosperm. Second one is the complete cell divide and redivide and form endosperm. It is called as a cellular endosperm. And third one is that is called as a HeloBL, HeloBial, in which the complete cell and nucleus divide and redivide and form a complex structure called as an endosperm. Remember this nucleus endosperm, cellular endosperm, and HeloBial endosperm. Later on, this particular endosperm undergoes division and redivision process and form a complex structure like this one, and here the endosperm develops over here. This endosperm later on is responsible for the formation of new plant like this one. So hence, this endosperm is the most important one. In this way, after the division, redivision process, a lot of changes are there, lot of division, redivision process are there, and where this endosperm formation takes place. Just see that here the quite a good example has been shown to you people that many polar bears are there, many ovules are there, and after a particular time, all ovules fertilize and all that form the endosperm. That endosperm later on form a matured structure, and it is called as a seed. This seed is the most important characteristic where the endosperm is present over there. Remember that if this endosperm divide into this one, then it is called as a polygonal type of endosperm that is present in the dicotyledon. And later on, if it forms the seed coat, then the seed coat separates from this endosperm. It is called as a non-endospermic seeds or albuminous seeds. And if, for example, in case of sunflower, you will find that such albuminous seeds or endospermic seeds, ex-albuminous seeds or non-endospermic seeds are endospermic seeds are present. Sorry. In case of jawar or maize, you will find that this endosperm dissolve in this seeds coat. So it is called as a non-endospermic seeds or albuminous seeds. In this way, this endosperm formation takes place, and this endosperm formation continues for a long time, and that results in the formation of seeds, and that seeds mainly are having two important type, that is called as a dicot and monocot. Just see, in case of monocot seeds, you will find that complete outer layer is present, and that outer layer is called as a seed coat, and it dissolves with this particular endosperm. So hence it is called as a non-endospermic seeds or albuminous seeds. Then in the central side of this endosperm, where the food material is stored over here, and in the central side, then epicotyle is there that cover the hypocotyle. Radicle is there. This radicle is responsible for the formation of new roots, and while from this epicotyle, that formation of shoot takes place, while towards the central side, the complete endosperm cotyledon is present. In case of monocotyledons, only one cotyledon is there. In case of dicotyledon plant, you will find that two cotyledons are there. So hence, this endosperm divide and redivide and form either monocotyledon or a dicotyledon. It is entirely depend on the characteristics that are present in a plant. So hence, this complete phenomena of formation of dicot seed and monocot seed is complete over here. You are supposed to remember this seed is structure, that is monocot seed as well as dicot seed, and how it forms. Just now you got the idea about it. So, okay, students, with this we are finishing about endosperm as well as seed formation. And remember the structure of the seed formation, because in theory as well as in practical, question may be asked on this particular one, that give the characteristics of the dicotyledon as well as monocotyledon seeds, how endosperm formation takes place, that also you are supposed to remember. With this, we are finishing about the embryology, and you are supposed to remember all this aspect of the embryology. And inshallah, in next class, then we'll proceed with the histology. Now we'll stop here. Take care of yourself. Thank you. Allah Hafiz.